All righty. All right, for those of you who are on Zoom, I'm not necessarily talking to everybody here. We'll get you started on this. But for those of you who are on Zoom, what you missed me talking about, and the reason I'm late starting this is because I was talking about how we have so many people that are missing class right now. And that part of, if you do end up being someone who is having to miss class, which if you're watching this on Zoom, you might be one of those people. And you have a reason for missing lab. So specifically lab, that missing lab attendance is something that is actually required by not just me or just my department, but the Higher Learning Commission. And they've actually defined that for my lab, for microbiology, you have to have in-person participation, and you have to have at least a certain percentage of it in person. So that's why we have that 6 absence rule. If you miss 6, then you will automatically fail the course. And things like doctor's notes and things like that don't excuse you from having to participate in lab. They don't. And so you still have to have that percentage of attendance. And that's why the lab days have to actually be physically in person made up. Also, the in-lab exercises that we do the uploads with, they have to be completed in conjunction with actually doing the lab. You can't just do them separately because part of that is activity. I know some of us that are sick at home or couldn't make it to the lab, that we would like be considering, like, I can just do this worksheet at home. But you guys will see, especially for those of you who are here anyways, for the lab that we're doing today, the lab we're doing today has gone over time for all of my sections. I just want to be clear about that. All we're doing is looking at your plates and describing what's on them with words that I give you the terms to choose from, as well as using microscopes. That's all we're doing. But we're going to be using microscopes every single day for the first half of the semester. It's really important that we learn how to use microscopes. So that's part of the reason why I can't just let you do that worksheet at home. Like, you have to actually use the microscopes. So I just can't. It just doesn't equate to the same thing. And so like I said, to people that were already here, we were describing it before we got started with this Zoom session, that one worksheet is 5 points, and we will have 18 lab worksheets that you'll be submitting. 5 out of those, 5 times 18, whatever that is, I'm not gonna do the math in my head. That whole in-lab stuff all together for all 18 of them is 5% of your grade. So just one of those getting left off isn't what's gonna cause you to fail the course. So if you're only missing 1 or 2 days, it's fine. But I already have some people that already have 3 lab absences. So you get 5. If you get to 6, you fail the course. There is no doctor's note that's going to save you from that. Okay? It's not my rule. All right. Okay. I don't know how to move this to the bottom. Oh, I can just do that. Okay. I'm gonna put this, whatever, I'll put down here. Okay. So I think everybody's signed in. I'm gonna go ahead and change my display settings. All right, let's go ahead and get started with our Kahoot. Don't worry, I'm going to share you guys back to this other screen. No, what's happening here? Okay, hold on. There we go. All right, let's go ahead and get started. So reviewing over chapter 5. Which of the following is found in a eukaryote, but not in bacteria? These questions are relatively easy. Sorry, that's not what I meant to click on. I feel like the first few questions anyways are pretty straightforward and easy. All right, so what do we find in eukaryotes, but we don't find in bacteria? A nucleus. So the eukaryotes, true nucleus, is really what that word means. And so it's actually in the name if you understand what that is broken down into. All right, next question for question number 2. This organelle turns chemical energy into cell energy. Pretty easy one. All right, so some mitochondria. Most of us are familiar with that one, so not too tricky. Next question. What is the function of the endoplasmic reticulum? All right, so the endoplasmic reticulum, it's the site of manufacturing of our biological macromolecules, including our proteins. So which type of endoplasmic reticulum do we see protein production in? The rough, exactly. Good job. Good job. What are the organelles that contain chlorophyll? Site of photosynthesis. All right, so chloroplast, pretty easy. They're what make the plant cells green or the algae green. All right, true or false? Yeast are fungi. All right, so this one, most of us got this one. It's going to be true. Yeast are actually fungi. So we have the two different kinds of morphologies of fungi that we could have, the yeast type and then the hyphae forming type. So obviously when we talk about microbiology and the fungi, we're mostly going to be focusing on molds and yeasts. And so the molds fall into that hyphae kind of group. The hair-like branching. All right, true or false? Fungi have chloroplasts and can perform photosynthesis. No. Oh, is it you? Yeah. Okay. All right, thank you. All right. Okay, we have chloroplasts. All right, so fungi do not have chloroplasts. They do not perform photosynthesis. We actually talked about how fungi can get their nutrients a little bit. So we had said that those fungi, we're going to have basically, what's the word I'm trying to think of? I can't. That's what it was. Heterotrophs. So they would get their nutrients basically from breaking down other things. It's what we do. We're heterotrophs. So we eat plants, we eat animals, we eat all sorts of stuff. And that might sound like, well, everything does that. Absolutely not. Plants don't do that. Plants get carbon dioxide that's inorganic from the atmosphere and turn it into organic molecules. And that's how they get their actual organic molecules built. They build it from carbon dioxide. So they're called autotrophs because of that. They are not heterotrophs. Anything that performs photosynthesis and uses that energy to fix carbon into an organic molecule is called an autotroph. And so fungi are heterotrophs. And there also can be saprobes, breaking down dead things, still not an autotroph. Or they can be parasites, so living off of living things, still not an autotroph, right? So those are the only things that the fungi will be. They will not be autotrophs. They will not be, and we'll learn more about autotrophs and that sort of stuff in the distinctions when we get to chapter 9. But we did talk about how they're heterotrophs, saprobes, or parasites. So they do not perform photosynthesis. All righty. Next question. Do algae help to provide oxygen to the atmosphere? So we know that fungi don't provide oxygen to the atmosphere. But what about algae? All right, so quite a lot actually. So we look at our choices here. The algae are our microbes that in this day and age provide quite a big proportion of our oxygen to our atmosphere. We also have like our cyanobacteria, which are not eukaryotes, right? And they perform photosynthesis also, but they don't have chloroplasts. They'll just do it on their cytoplasmic membrane. They have everything they need for it on their cytoplasmic membrane. So they'll provide oxygen too. But those microbes altogether provide our primary source of oxygen, and algae are huge contributors. All right, these single-celled organisms have trophozoite and cyst life stages. That reminds me. All right, so these are our protozoa. Matter of remembering that. But if we're talking about bacteria, we know bacteria can make endospores, but only certain species, right? So these would be our bacillus and clostridium. Bacillus and clostridium, okay? So the guys that make up our endospores, that can make endospores. And endospores are completely dormant or inert. They don't do anything, right? For protozoa, they make a similar structure. These are their cysts. Also dormant or inert. Okay? The active, multiplying, having metabolic activity and things like that in the cells, cells doing cell things. Those are our vegetative for our bacteria. And for our protozoa, those are trophozoites. Okay. So the vegetative is analogous to trophozoite, and the spore is analogous to cyst. Easy enough to say. We have fungi. Fungi make fungal spores. And these are only for reproduction. Asexual reproduction. So we're only going to be reproducing when conditions are good, right? So these spores are completely different from the endospores. And so that's why I think I mentioned previously in our last talk, if on the test I am talking about some kind of spores, I will say endospore, or I will be specifically talking about fungal spores. And those details will be in there. Most of you that tell me about spores, and then give you choices about bacteria and fungi and all this stuff, okay? So I will make sure that's delineated on my test. That's my responsibility there. But your responsibility to know that endospores and cysts are similar. The endospores are bacterial structure, that cysts are protozoan. Endosporozoan, these are for eukaryotes, right? So true nucleus guys. These are pro or eukaryotes, right? These are analogous to one another in that regard. And this is something else completely different. Same words. Because science is fun like that, and they like to keep us on our toes.
On that, you said that it's only only certain species, you said the bacillus
and clostridium.
clostridium.
Mhm.
So like with the bacillus, we know exactly what they look like under a microscope. They're going to do that, they'd be rods.
Exactly. So very astute observation. So that's one of those situations where there's something that's called Bacillus subtilis, for example, it's just a species of bacillus. You would know automatically that Bacillus subtilis is rod shaped because bacillus means rod shaped, right? And you would know automatically that it makes endospores. All the bacillus species will make endospores. All the Clostridium species make endospores. And those are the only ones that make the endospores. But luckily, the bacillus also tells you a little bit about their shape. One other thing that I want to bring up just briefly is that just because your name isn't bacillus, right? So let's say that you are Escherichia coli or Salmonella enterica or something like that, okay? that doesn't tell you anything about the shape, but can they be bacillus shaped? Yes. So not everybody's name says what shape they are, right? So with the endospores, bacillus has endospores, right? And its name is bacillus, so it has to be bacillus shaped. That's not going to lead you astray either. But Escherichia coli, it doesn't have endospores because it's not bacillus or clostridium, but it might be bacillus shaped. It's not in the name. You can't get that out of the name, right? I'd have to tell you that if I wanted you to know that. All right. Keep that in mind. Not everything has its shape or its arrangement in its name. And just because it's not in the name doesn't mean that it doesn't have it. Shapes and arrangements don't always work that way. All right, what are the helminths? All righty, so these are parasitic worms, which we talked about at the end of our eukaryote chapter. Our last question, what are platyhelminths? Obviously they're helminths, they're parasitic worms, but what kind? Excellent, that's gonna be our flatworms, flat. That is literally how I remember it. So with our helminths, our parasitic worms are called helminths, and they can divide up into roundworms or flatworms. The roundworms are called nematodes, and the flatworms are called platyhelminthes. And the platyhelminthes, the flatworms, can be divided up into two further groups. So those are going to be our flukes, which are our trematodes, and our tapeworms, which are our cestodes. So those are the nice, happy science terms that we have to know about these guys. All right. Yeah, something we don't want to think about, but it's actually really prevalent in the third world countries, and we'll talk about it in chapter 23. I believe they're roundworms.
Yeah, their eggs are.
Yeah, so I'm pretty sure that their adults are too. So they'll be classified based on the adult shapes. I'm pretty sure they're roundworms. I don't know enough about like the details since we do see, we'll talk about them, like they get briefly mentioned in the textbook, but there's so many that like different kinds of these guys. And we actually take it for granted here in the United States where we have very little, very few parasitic worm infections. There's actually
a lot of them in dogs.
Yeah, a lot in animals, in mammals in general. Yeah.
I'd say like a good 80% of puppies that come into the clinic will have some sort of one.
Yeah, and I feel like too with even people like that have dogs in like their own homes and everything like that. Like if you're letting your dog out in your backyard, and your backyard is frequented by critters like raccoons and possums and things like that, but they'll leave their feces or their partially eaten food or their urine or whatever behind in their wake, and those can be infested with worms. And you don't think about it, but if you're not regularly treating your dog with worm, anti-worm medications and giving them dewormed regularly, they're probably going to get worms. Like it's a high probability, not a small one. And some of those can get transmitted to people as well. So you could be putting your family at risk if you're not keeping your dogs and cats regularly, dewormed, especially if you're leaving, if you have an outside cat, your cat is gross. I'm just going to tell you, like, just guaranteed it's going to have health problems. So not me judging you, just stating a fact. The first thing we're going to do before we get into chapter 7 is talk about one critical thinking question. This one, we actually talked a little bit about it just a second ago, but a 36-year-old man presents with diarrhea and abdominal pain. Just a few hours before, he reports having eaten at a new Chinese restaurant. The only thing he ordered was fried rice, and he stated it tastes like it had been sitting under a heat lamp all day, so pretty stale. Eventually, after a few more cases pop up, the epidemiologist is sent to investigate. The epidemiologist discovers that the fried rice is contaminated with gram-positive endospore-forming rods. What is the most likely cause of the illness given this information and these choices? Yes. Yeah, exactly. So it's B. So we just discussed how endospores are only formed by Bacillus and Clostridium. That's the only clue you need from this. We've got on this screen a whole bunch, and it seems so easy now. When y'all reading these questions on a test, your brain is going to shut down for you because we know it will, because it does you those favors. And you're going to be having all of this information up here. And you're going to be like, oh my gosh, it's a lot. Where do I start? Well, the fact that I'm giving you that it is endospore forming, that automatically you should already book it down to Clostridium, which I'll put Clos, I don't want to write that whole thing out, this, or Bacillus, right? We know that their names have to be those. And we only have one of them up here. So we know it was Bacillus cereus just from that one little bit of detail. We do not need any other of the data that was up here to get to that answer, right? In the past, if I don't preface and give you this sort of a thing where I've told you guys, I reminded you that endospores are formed by Bacillus and Clostridium. We may have talked about it in the previous lecture, but if I just left it in the previous lecture and didn't remind people about it, most of the time people pick Salmonella for this because diarrhea, and people just assume that's got to be Salmonella. And I think I mentioned previously, there are thousands of things that can cause diarrhea. Diarrhea does not mean Salmonella, right? And so we know now we also learned Bacillus cereus can cause diarrhea. So we've learned already that stuff. Did you know that staph can cause diarrhea? Typically, it's not the staph infection that's causing the diarrhea, but the toxin that it makes. In fact, staph's toxin can live on your food. Like the bacteria will kind of live on your food, and it's not enough really to cause an infection in your GI tract. They don't survive in your GI tract, but they'll make toxins while they're on your food. So good old grandma brings some potato salad to your family reunion. She's made it and touched it with her love and her staph, and the staph will grow on there, and bacteria will not infect you. But the toxins that it makes are strong enough that you will get sick within 30 minutes to an hour. Most of the time, if you've eaten something and the thing that you ate made you sick within 30 minutes to an hour, it's staph toxin that made you sick. And it's extremely common. It's actually probably more common than the infective types of like diarrhea and vomiting and things like that. So that's called food poisoning. It is different than getting an actual infection and getting sick from it. Because if you were to get salmonella infection from, say, eating at back in the day, like Hardee's, when we used to have Hardee's, now we have Carl's Jr., but we had Hardee's, and the reason that it shut down here in Oklahoma is because we had outbreaks of salmonella. And you'll still see salmonella here and there. When you get undercooked chicken, you are handling eggs and not washing your hands before eating something or whatever it is. Salmonella will actually infect your gut, but you won't have symptoms until 5 to 7 days after. So you don't get sick the next day. You get sick a week later. So people are getting sick, and it's like clearly GI symptom related. And they're thinking, what did I eat yesterday? Oh, it must have been that place. It's probably a week ago. If you're actually sick with an infection, it's probably a week ago. People aren't thinking like that. So anyways, we know salmonella causes diarrhea. And we probably know E. coli can. Yes.
Is the staph one the only one that only lasts like 24 hours?
Yes, it only lasts 24 hours because once your body gets rid of that toxin, it's done. Like it's not making new, right? So it's self-limiting as well. And there's also no point in me giving you antibiotics because the bacteria aren't causing the problem. It's the toxin that's causing the problem. And there's no antibiotic. Antibiotics only treat bacterial infections. So there's no antibiotic that's going to attack the toxin. So you just have to wait it out. And of course, we know best treatment for diarrhea, fluids, and electrolyte replacement, and rest. So there's that. But that's pretty straightforward. If your diarrhea is persisting any longer than a few days, then and you aren't able to get fluids in your body, then definitely go to the hospital. But for the most part, most of these things we can kind of wait it out, and our body is meant to be able to work it out. But anyways, so other things that we got from this was gram-positive, and it forms rods. If we had just gotten that it was gram-positive and that it formed rods, that probably wouldn't have helped us very much because we could have eliminated Staphylococcus, because obviously coccus is not rod forming. So we could have eliminated that, but we don't know enough about Salmonella or E. coli really in order to eliminate them. So the real giveaway here was that endospore. Salmonella and E. coli, by the way, are both gram-negative and they both form rods. So then the only thing, and I don't expect you to know that right now, right? So that wouldn't have helped you here. So really that giveaway here is the endospore aspect. So these critical thinking questions, easy enough when we just talked about the thing. When you guys have all that information in your brain and you're ready for a test about anything, then sometimes that information can get a little bit tricky to navigate through. Read through your questions. If you need to write out the key points of what the information is giving you, and then look through your choices, be sure that you're reading all of your choices as well. And you're understanding why you're not selecting the other ones. Because sometimes if you don't understand why you're selecting one is right, and you're just going based off of word association, that can trip you up too. So be sure you're understanding what it's asking and why the answer is the answer, right? Easy enough on this one, pretty straightforward. On Tuesday, whenever I give the review, so I'm gonna be going over the 8 major topics for Unit 1, And I'll give you guys, trying to give you some variety of examples that you guys could basically base off of your own projects for 2, 3, and 4 on Tuesday. Yep. And so we'll start off with our typical review Kahoot for chapter 7. So we'll have that first. Then we'll jump into our 8 presentations that I will give as your examples moving forward, as well as review of those topics. And then if we have time after that, I will go over those 5 critical thinking questions that were in the announcements. And again, some of those are asking really basic questions, just kind of like this one was. It's just a matter of reading it and understanding it well enough to know that. Okay.
So this is the last chapter that's going to be included in our first.
Yes, this is the last chapter that's going to be on the test. On Tuesday will just be a review day. And then I have sent out that announcement that on Wednesday we're going to play games. So please join us if you have the time to do that. All right, if you don't have time to join us on the games, I think I said this in the announcement. If you don't have time to join in on the games on Wednesday at 4 p.m., then I can't record that. It doesn't work the same way. The recording doesn't work the same. It doesn't record the questions and the answers. It records just like the robot chasing people. You know what I mean? So that's what it records. Yes, the questions are on the phones. And so that doesn't get recorded in the recording. So that's why I can't record and post that. But I did post the questions and you guys can play through them and it tells you what, you know, when you get it wrong and stuff like that. So feel free to play through them the traditional way. There's no timer on them. I set it up so there wouldn't be a timer. So you guys could like work through them if you needed to, or whatever you want to do. So the review questions are the same ones. You can work through them the traditional way. But if you have time to play the game with us, that'll like test your ability to recall things fast. And hopefully, even if you haven't had time, hopefully you will have had time because that'll be the day before the test, right? Hopefully you will have had time to prepare some for the exam. But even if you haven't prepared really hardcore, it'll at least help you pinpoint maybe some topics that you need to focus more on, right? So come play the game with us, at least for a little while. It'll be for an hour, huh? They will be on Zoom.
Okay.
So there's a link that I posted. It's the same Zoom link as we always use. 4 p.m. on Wednesday via Zoom. And I'm just hosting it. But if you guys have any questions during it about questions that you might be getting right or wrong, just holler and or type it in the chat and I will answer your questions. All right, so moving on. Viruses. Viruses are acellular, right? So they aren't made of cells. So what are they made of? And we'll talk about that. But these guys are microbes that we know that if you are a cell on this planet, we know of viruses that can infect you. So they can infect anything, any existing cell ever, whether you're a prokaryote or eukaryote. There are viruses that can infect you, okay? So are they alive? What role did they play in evolution of life? We're actually gonna talk about that a little bit, not a whole lot, but a little bit. Their distinctive characteristics, if they're super small, and they are way smaller than you probably realize, and so insignificant, how can they cause such devastating effects? And what's the connection between viruses and cancer? So we're gonna get into all of that stuff as we talk about viruses and their characteristics. But first, let's talk about if they are actually like alive. So we consider them to be either active or inactive in the scientific community because we have defined cells as membrane-bound units that have everything that they need to make a copy of themselves right there in that one cell. And that's what we consider to be alive as well. Viruses don't have that. So they can sometimes have a membrane around them, but they don't have ribosomes. They don't have anything, endoplasmic reticulums or any of the structures that they would need to make copies of themselves or whatever. They do have a genome, but it's not always DNA. It gets a little tricky, but they do have a genome and they are coated in protein at the very minimum and sometimes also a membrane. And that helps protect them from the environment, but that's it. They don't have anything else going on in their cells or in their insides. So that's why we don't consider them cells. And they're going to use your cells, the host cells, machineries and resources and nutrients and everything to build copies. So sort of like aliens, so like they are literally a parasite. So that's where we have this term up here, obligate intracellular parasite. They're obligate, that just means obligated. So they have to be that thing. So they're obligated to be intracellular, so inside the cells, and parasites because they're causing damage and using the host resources and stealing things from you. So they'll use your cell machinery to hijack everything that your cell is doing and take over pretty much every single process in your cell and direct your cell to making virus copies. And that's it, that's all they do. They go into a cell and take over that cell to make copies of themselves. And those copies move on to new cells and they take over and make copies. That's all they do. And so how does that lead to such devastating disease and effects? Well, we're going to talk about how. But they're not cells, and they're very, very tiny, and they're very, very basic. All right. Viruses have influenced sometimes our genetic makeup. 40 to 80% of your genome is virus left over from past. So you're like, from me having, no, from I mean like ancestors, like back from like caveman days and all of the ancestors leading up to now. Viruses can actually, some of them, not all of them, some of them can actually incorporate their DNA into your DNA. And that can even be spread from ancestor to ancestor over time. And it looks like a lot of our genome may have come from viruses. Sometimes they could confer traits or new genes to us that way, and sometimes not. And we don't know enough about that viral ancestry to really know much about it at this point. We're still, that's one of the big topics in like genetic investigations that they're doing, try to figure out which of this came from viruses, which of it didn't, and then how that led to development of man, and how many of these are genes that are actually active, because some of them are actually useful and active. They're not just trash, okay? So that can actually be really important. And I will tell you what, viruses are extremely important in helping bacteria become infectious and deadly and dangerous. So a lot of bacteria actually get their genes for their toxins and things like that from the viruses that infect them. So that's something else. Now we're talking like meta level of infection, right? Bacteria that were infected by viruses, and those bacteria infecting you. Now, what if I tell you there are bacteria that are infected with viruses? Those bacteria can infect parasitic worms, and that because of that relationship, the parasitic worms can cause disease in you. That's too much. A multi-level parasite. But yeah, it's true. So we're going to move on to size comparison. This is a yeast cell. Yeast are pretty small eukaryotic cells. They're kind of, I say, kind of average size compared to maybe some of the cells that are in our own body. But this is one yeast cell, and this is the daughter cell budding off of it. So two separate cells. This is E. coli. This is strep. Okay, these are bacteria. This is a bacteria called rickettsia. It's like what causes Rocky Mountain spotted fever. So there's some viruses. So this is a virus here, and this is a virus here. Mimi virus and Pandora virus. These are the largest viruses that we know to exist. And we're going to talk about them too much because we're not worried about them causing disease in people, but they exist. And these guys are big enough to be bigger than rickettsia, but still not as big as like the typical bacteria, right? So there's some viruses: herpes, rabies, HIV, flu, adenovirus. This is a T2 bacteriophage. A bacteriophage is a virus that infects bacteria. Okay, so this is a virus that's small enough to infect this guy, E. coli specifically. It's an E. coli infecting bacteriophage. This is poliovirus. This is yellow fever virus. And this tiny dot down here is just a hemoglobin molecule. Not your red cell, which would be about the same size as the yeast cells. Just the hemoglobin molecule. Just one molecule of hemoglobin. And look at it compared to the size of yellow fever virus. That's pretty crazy. So it's one protein molecule compared to a whole virus. So these smaller viruses like polio, yellow fever, or parvo, like canine parvovirus. These guys are super tiny, super, super tiny, but super, super basic, but also can have massive effects. You're going to tell me that polio doesn't affect you? Go ask FDR. Well, ask him when he was alive, but you know what I mean, right? So we talk about causing permanent paralysis or even progressive damage. You can get over polio and still have the damage like continue after you've cleared the virus, degrading your muscle ability to function and everything like that. Go ask the people that lived for years in iron lungs. We just lost, I think last year, the last person that was in an iron lung because of polio. He did a lot more with his life than I will. I'm not gonna lie. He got like PhDs and like did all this stuff. Yeah. Well, so potentially yes. So interesting questions.
Question that is, so with herpes simplex, like there's two different viruses.
Yeah.
So with the herpes simplex, like they estimate that like I think they said I read a study that says like 70% or more will happen some of them in the year that they have it. Is that genetic? And that's like our my say like one of our parents had herpes simplex virus, would that virus then be able to was based off of it, hormones might have gone through.
So usually not. So they don't usually take up home in, so that would require them to be taking up home in like your gametes, right? Like your egg cells or your sperm cells. And those usually aren't conducive to most of those viruses living. So in order for that to happen, those would have to be, so most viruses aren't heritable like that. Most of them live in like what we call your somatic cells, like your body cells, but not your reproductive cells. We don't see a lot of viruses that actually get transmitted through the reproductive cells. That's something that I think honestly has just kind of gone away throughout evolution, and we just see some of the genes left over as a result. But that becomes a little bit more complicated when we start looking at how that works with like old viruses. But the viruses that we see nowadays, especially all of the ones that we'll be talking about in our class, and all the ones that you probably traditionally be familiar with, they aren't going to infect your gametes, and so they wouldn't be heritable through genetics. Now, but there is an issue, of course, if you have herpes simplex. There's two types of herpes simplex, like Shai was saying, herpes simplex 1 and herpes simplex 2. Herpes simplex 1 is traditionally associated with oral herpes. So we're talking about cold sores. That's why we're saying 70% of people having it, because if you've had a cold sore, you have herpes simplex 1, okay? But it can also be transmitted sexually. So that's where some of that gray area comes in. Herpes simplex 2 is traditionally sexually transmitted in the genital area, but it can be oral as well. So again, some crossover there, okay? But regardless, once you have herpes, herpes is forever. That's not just saying, it's a fact. And once you have it, it hides out in your cells and can be expressed whenever you're having stressful moments. Medications like Valtrex can help suppress your body from making new virus, but you will always have it. And so that can help with the expression and the symptoms of the herpes. You can still technically be contagious with herpes even when you don't have outbreaks that are visible. So that's something to be aware of. You're not 100% free of risk of transmitting it to people. So that's something to be aware of as well. But the biggest risk for herpes, especially with herpes type 2, would be what we call vertical transmission. So it's transmission from the mother to the child during birth. And so if you have herpes 2, herpes simplex 2, and you are about to give birth, if you have any evidence of a recent or coming outbreak or active outbreak of herpes, you are not allowed by law to give birth vaginally, because the risk of your child dying from that herpes infection can be as high as 80%. So we're talking low end 30, high end 80% chance of mortality for the baby being exposed to that herpes. That's not small. And so even if you're like, well, I'm having a vaginal birth, now you're being selfish at that point, when we're talking about a 30% mortality rate. Yes.
Is that because it's contact?
Yes. So it can be contact related, and I want to be very clear about that. Just because you're having an outbreak like on your inner thigh, even if we keep that baby away from the inner thigh, there could be virus that had transmitted to other areas in the region during that active outbreak, right?
So if there's not an outbreak,
it's usually fine as long as you haven't had an outbreak. There's no evidence of you having had an outbreak within a certain time period. Now, yes.
so there is an outbreak, you may still be like seen after.
Yeah, as long as it's not in that area. Yes. So that area is the risk with herpes. It's always that area that we have the actual outbreak. And with that thigh area, as I'm sure we know, as people, like you have an outbreak on one thigh, that whole area is kind of like at risk for contamination. So we'll still give birth to by a C-section. You still give birth. We'll have the baby by a C-section, and you can still have skin-to-skin contact afterwards. Just want to keep it away from any outbreak areas. The risk there is going to be contamination through the eye area, which is actually not uncommon. And then if it gets into the eye, we risk meningitis caused by herpes, which is that's where we start getting into that 80% mortality rate area. Makes sense, right? And then once that baby has been exposed, because they can be born by a C-section, even if you're having an active outbreak of herpes in your genital area, you can be born by a C-section and not get herpes as a baby. You won't have it. But if you're exposed to it during the birth, then you'll have herpes, and you'll not just have it as a baby, you'll have it forever for the rest of your life. So
like with type 1 and type 2 would be, like if we were testing for that much scientific, I'm thinking we could distinguish the two using like a selected.
yes, you can. So there's like PCR tests and things like that. So PCR, for those that don't know, we'll learn about it in more detail later, but it's based on the DNA, so the actual genetics of things. So whatever the organism is. But you have to know what genes that you're testing for. So you can't just take a DNA sample, put it in a machine, and the machine just tells you without any input that this is genetically this thing or that thing. You have to give that machine some information. And so it'll look for certain genes using what we call primers, and we'll learn about that later. But you have to give it those primers in order to know to look for this gene in COVID or this gene in HSV1 versus HSV2. So those sorts of things. So we don't always have primers like that available in all hospitals, like things that are pretty common like herpes or COVID or something we do. Most bigger hospitals are going to have that. But if somebody comes in with an exotic disease, you can't just take a swab of their goo and put it in a PCR and it tells you that it's that exotic disease. You have to have the primers for that. So that's where the testing for these things can get tricky.
So the difference, so HIV, I read something that said.
Mhm. Yeah.
Is that because it's,
So
or is it-
that's because of blood. So that's because of blood exposure. So they actually say a lot of times too with sexual transmission, HIV can be transmitted through semen, but it's actually much higher risk when we're talking about blood exposure. So it can be transmitted through semen, but the way that it gets into, like, let's say it's a male-female sexual encounter, the way that it gets into the female isn't just because it's in the semen and it just gets in. It's because it gets in via micro tears in the vagina during the sexual practice. And so there's some blood exposure risk there just from the act of having sex. So it actually is a bloodborne and sexual fluid-borne thing. And you can transmit HIV through like breast milk, for example. It's very low incidence, but it does happen. You do not transmit HIV through saliva, for example. But there's risk for all that. But it is bloodborne, and it is present in like pretty much once you get HIV, all your T cells are going to be infected with HIV. And it becomes incorporated in your DNA and your cells, and there's no getting that out, which was mentioned previously. But yes. So the post that I made on Canvas about that was that I had a discussion with one student was like, I had mentioned that HIV is 100% fatality rate. And they're like, no, people live their whole lives with HIV. What are you talking about? That's not 100% fatal. When you're on medication, yes, but you stop that medication. You just, even if you are, Magic Johnson and he's been having HIV forever, he's been undetectable for forever. Right. You stop him on his drugs, those viruses are going to start being expressed again. He will eventually develop AIDS and he will die. 100% chance of it. Not a small chance, 100% chance of it. So it's only the drugs. Sorry, what?
Other people die from complications before dying from pneumonia and stuff like that.
Right. And so it's people don't realize that it's not the actual virus that is killing you. It's not the HIV virus that's killing you. It is the AIDS. And the AIDS is the lowering of your immune system. And by having AIDS, you're more susceptible to these other infections. And those are usually the things that kill you. But we're talking about those other infections being things that healthy people will never have. Know that they might have viral expression. Yes, and it's affecting their T cell counts, and your T cells are part of your immune system and what keeps all that in check. Were you gonna say something?
And more of a question.
Uh-huh.
So like in like our modern day, what is there or like what is the like if you get this like I know rabies is like 99.
Rabies is 100% fatality, right? We have had people survive it in a coma forever.
Right.
So it's like you want to sign up for that. But that's also really rare. So rabies, that's why we say rabies 100% fatality rate, because what kind of life is living in a coma if that is how you would survive it.
Right.
But yeah, that's the only instance we've had people actually survive rabies. I want to be very clear about rabies too, okay? Rabies is easily, it should be, if you're sitting here being like, yeah, whatever, rabies, whatever, it's fine. We have treatments for that. You do not have treatments for that, okay? I want to be very clear about this. You get rabies and you get bitten by an animal that is rabid. Once you have symptoms, and it does take a while, okay? You're not gonna have symptoms like a week later, but once you have symptoms, it does take about a month, 3 weeks to a month to develop symptoms from rabies, from a rabid animal, okay? You start getting symptoms, it's gonna start like a headache and a fever and feeling crappy. Once you have even those minor symptoms, there's nothing medicine can do for you in the entire world. You are 100% gonna die.
That's what it is. Even if you don't know.
So if you get bit by an animal that you suspect is rabid, you can go to the hospital and you want to catch this within the first week, minimum. So that's why they say if you get bitten by an animal that you think could potentially be rabid, you need to go straight to the doctor immediately. Your kid gets bitten. Like, I was playing with the kitty out in the street, LOLZ, it bit me. And you don't just wash that kid's hand and send them back to school. You go to the doctor because you don't know where that cat's been and what kind of rabies it may have had, right? Anyways, it's harder with cats and like dogs that look like they're domesticated because they'll be out in the daytime and you don't always know. And like they might bite you and you might just think, oh, whatever, cats bite because they're nasty. I don't like cats in case you can't tell, but I'm not a cat person. Anyway, so they might bite you and whatever. But like if that saliva gets into the body, rabies can infect pretty much any mammalian cell, which is also why possums can't get rabies. They're not mammals. You guys didn't know that. They're marsupials. Okay. Yeah, they can't get rabies because they're marsupials. So I think they're the only marsupials in the northern hemisphere. They can get plenty of other diseases, please. They can get leprosy, for example, but they won't get rabies. So you don't have to worry about that.
Don't worry, your pet won't.
You might get leprosy. But anyway, so you get bitten by a raccoon, or you get bitten by a coyote, or you get bitten by some wild animal of any kind. That saliva gets in your body. If you go to the ER, you go to the doctor, and you tell them they're bitten, they will give you immunoglobulin that is against rabies. So they'll harvest this from people that have seen rabies or have immunity against rabies. It's very expensive, really expensive. Okay. But they'll give you that immunoglobulin, and what that is antibodies that are made against rabies by some other human being, and it will help protect the virus from getting into the cells where you're going to be screwed, right? So it protects you from that, and will also give you a vaccine so you can build up your own antibodies at the same time. And they will do this administration of that vaccine into your gut because that's where most of your immune cells are. This is not a fun process, and it's not something that you get done and then you get 2 shots and you go home and you're fine. It is also extremely expensive. It's also why anybody here work at a vet clinic? How many of you guys are vaccinated for rabies? Okay, lucky. And you probably know you're lucky because most vet clinics won't vaccinate you unless you are like one of their primary workers. And I'm not kidding. So rabies, usually, well, like I'm not kidding. A lot of vet clinics don't vaccinate like their traditional like techs and things like that because it's just expensive to do it, and they're not keeping you on typically for long enough that it's actually cost effective for them. But rabies is actually more common than you probably think it is in the wild animals. Okay, in our domestic population, most of the time, people are pretty good about getting rabies vaccinations. And even though we recommend that you get them every 1 to 3 years for your pets now, it should be every year, right? I still want you to do it every year, but it's about 1 to 3 years now. The vaccine really does last quite a long time and can last for, I don't want to say indefinitely, but like 10 years. Okay, so, but they're so adamant about keeping up with that and making sure you're doing it that they want you to do it every year for your pets, because if it gets out to the human population and it affects one person, I mean, imagine one little kid gets a tiny little bite from a cat and then you're going to die from rabies. 100% fatality once you have the symptoms. But if you can make it to the doctor within that first week of exposure, we can give you all these expensive shots and treatments. You are not going to find that your insurance will pay for that, I bet. But your insurance is going to find you negligible.
Only thing that can save your life.
Right. Your insurance is probably going to say you shouldn't have been around, stray animals.
Why didn't you test for rabid animals?
Right.
Yeah.
We see a lot of like stray dogs in like Mexico and areas like that where they are actually probably rabid. Like you can almost bet on stray dogs that are in like Mexico City roaming around. We have this issue a lot in like India, in places where we have a lot of stray animals that might seem otherwise friendly, but here, there, interspersed with them, we'll have these rabid animals. You get one bite from them. If you ever see, by the way, a raccoon out in daylight or a coyote out in daylight, they are probably rabid. Just bank on it. Here in the US, well, here in Oklahoma, we have big issues with skunks and foxes. So skunks and foxes, but all around the US, skunks, foxes, raccoons, coyotes, pretty much if it's a mammal, it can have this. Bats are a huge contributor to the rabies problem as well. Because they can live in your house and then like get inside from your attic and then like bite you and you didn't even know it while you were sleeping. Yeah, especially for pool. Because the mosquitoes and the bugs that hang out over the water, yeah. Anyways, so we're definitely going to be running a little behind on this lecture. So viruses. Viruses, the most simple viruses are going to be like this guy here, okay? This guy here is what we call a naked virus. And what he is the absolute bare minimum of what all viruses need. It's important that you remember this because it will definitely be on the test. The 2 structures that all viruses need are their protein coating, which is called a capsid. Okay, capsid is spelled like this. All of them have to have a capsid, And all of them have to have a genome, which is the squirrely guy on the inside, okay? Looks a lot like our DNA double helix. Sometimes it is DNA. But I want to be very clear, just sometimes it is DNA, okay? So it can be DNA, it can be RNA, it can be single-stranded, or it can be double-stranded of either of those. In fact, probably me saying double-stranded RNA already to you guys could sound a little weird. If you know anything about genetics, and we know we have double-stranded DNA, it's the double helix, the two sides that twist. When we make our working copy from our DNA, our genetics go like this. Your DNA, who uses these markers? And I don't know. Your DNA becomes messenger RNA. We know our DNA is double-stranded. It turns out your messenger RNA, your working copy, that working copy of that gene, that messenger RNA, is single-stranded. And then ribosomes are the machines that'll read this messenger RNA and turn it into protein. Talk about how that works in chapter 6. But this is the gist of it. And this whole DNA to RNA to protein setup, we call the central dogma of genetics. All right. So some of our viruses can have a single-stranded RNA as their genome. This is great because then they'll just stick that single-stranded RNA into our cells and our ribosomes can just start making protein from it. We don't have to wait for it to be turned into RNA. Obviously, if they're a DNA virus, all they have to do is turn that DNA into RNA and go through the central dogma like usual. Some of these guys are double-stranded RNA. Then all it has to do is separate those two strands. And we have one strand we can make copies of RNAs from, and one strand we can just start reading right away. So we would have two strands. One strand of DNA is what we use to make our copy of our RNA, right? So our two strands of DNA. We have one strand that would be used to make the copy of the RNA. We would call this the template strand. The other one is called the coding strand, and it has the same sequence as what your RNA would have. It's just the DNA version. Because you remember A to T and G to C and C to G and so forth, so on and so forth. So the A's binds with the T's and the C's binds with G's. So if this code read A, C, C, G, this one would read T, G, G, C, right? So this, where it would have been on our DNA, wow, I don't know. It's barely half of you can even see it anyways. If our DNA, I'm gonna put D, okay, said A, T, C, C, then your RNA that we're making the copy of for our working copy would say U, A, G, G, right? And then we can run off with this messenger RNA single strand and then use that to make our protein and read it. But this one that we're using as our copy, our version to make our copied RNA from is the template strand. The coding strand reads the same thing. It just is the DNA version of it to keep everything double-stranded. So we have our nice copies. We're making copies of cells. Sometimes one strand is our coding strand. Sometimes the other strand is the template strand. It just depends on the gene that you're looking at. And that's where it gets really complicated. But it's a little important just to understand this because you can have double-stranded DNA viruses. You can have single-stranded DNA viruses. You can have double-stranded RNA viruses. You can have single-stranded RNA viruses. And those single-stranded DNA or RNA guys can be positive sense or negative sense. This mRNA right here that we can run off and read with our ribosomes, this is positive sense. If we needed to make a copy of it with our ATCG rules, if we needed to make a copy of it before we could read it, then it would be negative sense. So negative sense needs a copy made before we can read it. So what's the point of even having a negative sense? Well, just to make more copies. So you have a negative sense there and it just serves as a template. You can just crank out positive, 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 positive. And that's when we have a ton of them for your ribosomes to read from and just make protein, 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 and start making the results of that. Okay? So that's the genetics of that. So I just told you viruses can have 6 types of genomes. You have double-stranded DNA. Bacteria have double-stranded DNA. Archaea have double-stranded DNA. A virus, depending on what species it is. Some viruses have double-stranded DNA. Some of them have positive sense single-stranded DNA. Some of them have positive sense single-stranded RNA. So it's 6 different ones. Let me just write them all on. Yeah, it's a lot. So I'll write them up on the board. Maybe. This one, we're going to get to the questions, so let's be real here. But you can have single strand or double strand. You can have positive or negative of your single strand, and this could be DNA or RNA, and this could be DNA or RNA, and then this could be DNA or RNA. Whatever, you get it. I probably could have written this a little bit better, but anyways, you get it. So there's
They're
6
complex.
different ones, okay, that they could have. Now, if you are HIV, for example, HIV always has positive sense RNA genomes. It always has positive sense RNA genomes. That virus, that species of virus always has that one genome. Like it can change it up. So whatever species of virus you are, you have that kind of genome, always. Okay. Now, maybe that's starting to get you to be thinking, I just told you that HIV has an RNA, positive sense RNA, so just like our mRNA. HIV has positive sense RNA, single strand genome. So when I told you that it inserts DNA into your DNA, it's not RNA that's inserting into your DNA. So when I told you this is the central dogma, and before we discovered HIV, we didn't think anything could ever make DNA from RNA. We thought that was a travesty. HIV defied that rule. HIV carries an enzyme with it that helps it turn its RNA into DNA. This process right here is called transcription. We're just put transcript. Rewriting, basically what that word means, right? So transcription, HIV contains an enzyme called reverse transcriptase. ACE means enzyme.
That's right on the nerve.
Yeah, it is.
On the nose.
Right, so you kind of know already what it does, as long as you know what transcription is. But reverse transcriptase turns its RNA into DNA. Great target for a drug, don't you think? And it is. But once that DNA gets put into your cells, you're not getting it out. You can target reverse transcriptase with your drugs, and we do. And it'll prevent you from making new viruses. But what's already incorporated in your genome, we can't get out. So PrEP and drugs like PrEP and the drugs that we use to actually treat HIV infection target reverse transcriptase and other genes that HIV has that help with this process directly. But it's not enough to get it out of your genes once it's there. So PrEP is great for preventing from getting HIV because it can't make viruses and things like that. And it can't turn its RNA genome into DNA, and you can't get infected. It can't get inserted in there, and you're safe if you stay on your PrEP. Still highly recommend if you are taking PrEP and you are engaging in sexual activity with someone who is HIV positive that you still use protection, even if they're undetectable, right? Like I said, once you have HIV, you have it for life, for now, right? All right, so you have to have a protein coding and you have to have a genome. That protein coding is called that capsid. Capsid, protein shell that surrounds the nucleic acid. Those structures together are called a nucleocapsid. Sometimes you can have your nucleocapsid, your protein around your nucleic acid, right? And you can have that protein nucleic acid combo. And then when you're leaving a cell, you'll just imagine the cell's envelope is like a big blanket around the whole cell, and you're a virus that's on the inside, of the cell contained by this blanket. And you walk out of that cell and you just surround yourself with that blanket, and you walk off with a little bit of that blanket just wrapped around you like a Snuggie or something. That's how these guys get envelopes around them. So the viral envelope is the double membrane, that double lipid bilayer membrane from the host. They just wrap it around themselves and they're
They
leaving.
just say, oh, thanks.
They just take it with them. So,
So they don't make anything for themselves?
no, I mean, they just use our machinery to build everything. And then they have these things called spikes. Yes, so our spikes are our keys into your target cells. So think of it as you're carrying around a key that will let you into human liver cells, or a key that will let you into pretty much any mammalian cell, or a key that will let you into any primate, lung cells or something, okay? So here's an example of our naked virus. A naked virus is just the capsid and the genome. Here's our genome. And then we have the capsid coating. The spikes in this case are part of the capsid, right? So there's more proteins sticking out of the proteins. For our enveloped virus, where we stole some membrane, we had to stud the membrane with our keys, our spikes. Because if there's part of the protein part and we're getting out with the membrane, we can't actually use our keys, you know? So if they're gonna put keys on themselves to get into the next cell, which they have to, those spikes, those keys have to be on the outside of the membrane. So what they do is they completely take over your entire membrane. Any of the normal proteins that are involved on your normal outside of your membrane, which you might be sitting here thinking like, okay, well, so a few of them, anything that controls allowing you to move things in and out of the membrane, anything that allows you to attach to surfaces, to attach to the cell next to you, to engage with the cell next to you, to receive signals from the cell across the way, to recognize that this is a self cell. Any proteins like that on the surface of your cell that perform those functions, gone and replaced with spikes in these viruses. So that when they leave, they can leave through that part of the cell and just cover themselves with their pretty blanket with the design that they put on it made out of those spikes. Okay, so now you're starting to understand why your cells treat you poorly when you're sick with a virus. They can't stick to the cells next to them. They can't tell the cells next to them that they are self. They can't do anything that they normally would that way. So when we start thinking about Ebola, Ebola is very effective at this. Ebola replaces those surface proteins so well, you can't adhere to the cell next to you anymore. You also, because if I am a cell and this is the cell, right? And I'm infected and I have the key to get into this cell all over my surface because I'm getting ready to make viruses. They're going to bud out and cover themselves with blankets that have the keys to the next cells on them. I'm a cell that has those keys on me in the meantime. So I start to just naturally, because they're just proteins doing protein things, be able to open up the cell next to me and merge with it and become one with it. Just like the viruses would do when they start to get into our cells. Now our cells start merging with each other, making these big old giant multi-nucleated, multi-cell all under one big old membrane. That structure and that thing that happens is called a syncytium when it's one. And we have multiple called syncytia. Anybody heard something like that word before? RSV, respiratory syncytial virus. It's in the name. So they're merging their cells next to each other just because they're covered in these spikes. With Ebola, we'll do that. With Ebola, we'll also detach from our surfaces that we're attached to the point that first your capillaries become weak. Your cells that make up your capillaries can't make up capillaries anymore because they're just detaching from one another because they're infected. So just start just detaching. Suddenly now, instead of containing your blood, we're leaking our blood out into your tissues. And eventually we'll have so much spread of that virus that cells are just falling off all over. You start bleeding out of your eyes and your nose and orifices that you didn't even have before. Now your cells are falling apart and your body's falling apart and you're just bleeding out everywhere. That's how that happens. Just from some proteins. That's how viruses are so nasty, okay? We have two different kinds of capsids that you can have. The helical capsid, where did it go? There it is. The helical capsid and the icosahedral capsid. The helical capsid is as follows. Our little tiny units that make up the capsid, the whole protein coating is the capsid. The little units that make it up are called capsomeres. Those capsomeres circle around the genome. Like if this was the genome, the capsomeres build up around it helically until it's surrounded. And that can be enough to make a virus. But also we could take that surrounded thing and just ball it up and then push it out through a membrane. And now it has an envelope around it, right? So that's how Ebola works. And so they can be round after that. They could maintain their rod shape after that and all sorts of stuff. So it's actually more common to have, most of the viruses that we talk about will be enveloped, but obviously we have plenty of them that are naked. That can cause plenty of disease, right? So those are our helical guys, our icosahedral guys. We get our little tiny protein subunits, our capsomeres, and we'll put them in triangle shapes and we'll just stack up those triangles in an icosahedron. I don't know how to describe it other than that, but it really is how they will organize themselves, okay? And those can be enveloped or non-enveloped as well. Next, we have the bacteriophage. These are viruses that infect bacteria, okay? So phages are viruses that infect bacteria. I usually just call them phages. They're pretty typically called phages. But more specifically bacteriophages, okay? That's what they look like. So this is, of course, the artist's rendition of it right here, but this is an actual electron microscope image of one. This is literally a picture of it. Tell me that's not weird. So these guys have their icosahedral head that contains their genomes. That's their like true capsid. But these other protein structures that come off of it, the little feet, the legs that we have there are basically their spikes. Okay, and so those legs will recognize, oh, I landed on E. coli. When it lands on E. coli and the keys have recognized the locks and we start unlocking, we'll squat down, shove the tail pins into the cell, squeeze down that sheath that looks a lot like a spring, squeeze it down, and while we do that, inject the DNA straight into the E. coli cell. So viruses that infect bacteria never enter the cells. They just inject their DNA into the cells. Okay, and then the other side of it is viruses that infect bacteria, when they multiply, they just multiply until they burst the cell open. Our viruses don't do that. It doesn't do them very much justice. It would kill us very quickly if they did that, and they would be less likely to be able to spread and things like that. Most of the time, our viruses will either bud out, which is whenever they cover themselves with that blanket, and they'll just pinch off the membrane behind them as they go, and our cells are left relatively intact. Or our cells will expel them naturally through a process called exocytosis. So exo outside, cyto cells, process of getting them outside the cell. It's sort of like when we engulf something and eat it up. It's like the reverse of that. Okay, making them in vesicles from the Golgi apparatus probably, and then just, kicking them out. That's exocytosis. So luckily we don't burst typically. All right, so that viral envelope, like we said, if you're covering yourself with an envelope from the membrane from your host, you're going to want to replace your host proteins with the spikes so that you have your spikes on your outside. The spikes are usually glycoproteins. Just proteins that have some sugars involved in them. Just helps with that, allowing us to make unique keys that could fit into the locks. They mimic normal molecules that would normally be associating with the surface of your cells, and they mimic that. Just allow them to get into whatever cells that they're targeting. Okay, some of them have a lot of genes, other ones have like just a couple genes. So it just depends on the virus. Also, if you are envelopes, those can be our more complicated ones. They can also carry other things inside of the virus with them. We've already talked about positive and negative sense. We also talked a little bit about reverse transcriptase. So all the viruses that can do that, where they are mRNA and they can revert back to DNA, we call them retroviruses. So retro, like going back in time. So retroviruses can do that. They'll also borrow some of our other resources like tRNA. We'll talk about what those do in future chapters. All animal viruses go through the following steps, I guess. There's 6 steps for animal viruses. Okay. Adsorption, penetration, uncoding, synthesis, assembly, release. So adsorption, this is attaching. Penetration is getting in. Uncoding, remove your capsid, so you can release your genetic stuff, and we can start doing things, making copies, right? Obviously, that's what synthesis is. We are making copies of the genome as well as making our proteins to make up our new capsids, right? So we'll make our copies of our parts with that synthesis. Parts. And then obviously assembly is putting those parts together. I really wish I had my little writing pad. I should just steal it from the other room. And then release, that's pretty self-explanatory. After we've assembled the thing, we need to get it out of the cell, right? We talked about 2 different ways that you could get released. The 2 different ways is by budding. This is the biggest together I've ever written. But you can be released by budding or by exocytosis. Okay. So that's the steps involved in animal cell infection. We good? All right, so this goes through the details of that. I'm going to move ahead, just touch on things where I think it's important. Obviously, if your key only lets you into using your key only sees the lock, that we only see that lock on human liver cells, you can only infect human liver cells. So if your key sees the lock that's found on all intestinal and nerve cells of all primates, it's a little more general. And if your key sees the lock of pretty much any mammalian cell, then that's really general. So that's like what we see with rabies. All righty, penetrating, we kind of already talked about this, but basically your key sees the lock and you unlock it, and you will either be eaten up by the cell, by phagocytosis or endocytosis. Endocytosis, we're not just recognizing you as just junk and eating up to digest you. Endocytosis, we have you literally used the correct key in the correct lock, and the cell's like, yes, please come in. Let me let you in to do your job, because we think it's something else. That's endocytosis. And then we have this other version where you can see on the bottom picture, we have used our key in our lock, and now we're going to merge our blanket back in with our blanket of the new cell and just shed the blanket while we were doing that. And then that empties our contents into the cell that way. It's a little bit different than endocytosis, it's just proper uncoding. Either way, you're going to have to lose that protein coat to release that genome so we can start making copies of the genome and whatever protein products we need in order to build that. If you are an RNA virus, we can do all of this in the cytoplasm. We can make copies of your RNA in that cytoplasm. If you are positive sense, we will make a negative sense so we can build more positives from the negative. If you are negative sense, we'll make positives from the negative sense that we can read the positives and then use the positives as templates to make more negative sense that we'll package up. Okay, things like that. We'll also be making our products from our proteins like the capsomeres and the spikes. We'll stud that whole outer membrane with our spikes, push our way through it with our nucleocapsid, and coat ourselves with that envelope. If you're a DNA virus, everything associated with DNA happens in the nucleus. DNA doesn't ever leave our nucleus. So if you want to use our machinery that's associated with DNA stuff, you have to go in the nucleus. So DNA viruses have to do everything in the nucleus. Okay? Here's a picture of a nucleus where we have a virus that's infected. This whole blob here is the nucleus, and everything on the outside is the actual like cell with the cytoplasm and everything. This crystallized structure here is just proteins of new viruses. We've just made so many proteins from this, the crystallized and like stacked up on each other. And all these little dots in here are new viral particles. See how it takes over the cell, just like, and you can see this by looking at it in a microscope. Anyways, we talked about exocytosis and budding. Cytopathic effects are things that you'll be able to see in that microscope, whether it is making syncytia, so joining our cells together next to us, or detachment from the cells, or the cells might not understand like what's usually on the bottom of the cell attaching to other cells, what's usually on the top. Like if you have intestinal cells that have villi on them, they might not realize that the villi need to be going on the outside. They can't tell what's outside or inside anymore because that's usually determined by their surface proteins. So when we start having cells misbehaving or doing things that they normally wouldn't do, these are called cytopathic effects that we see as a result of viral infection. So that's going to be obvious things that we can actually see with microscopes. Some of these guys, like we said, can hide out in your DNA. They can hide out in your cells for extended periods of time and not be shedding virus. When they get incorporated in your DNA like that, it's not part of your DNA. It's not your genome. It's still the virus genome. That's called the provirus. That leads to chronic latent state. Then we have oncogenic viruses. So anytime that we have genes that the virus makes that disrupt your cell cycle, or they will have like incorporate themselves into parts of your genes that disrupt your cell cycle, or they have proteins and other things that can get involved in that stuff too. But that basically leads to your cells growing unchecked. And so where we normally would stop them and say, hey, you're doing something wrong genetically, or you aren't supposed to be multiplying or whatever, that stuff goes out of control and leads to the development of cancers. We estimate about 13% of human cancers are caused by viral infections. We're probably more than that, but that's the ones we have evidence for. And this includes HPV, which is human papillomavirus, which is why the whole reason why females are supposed to be getting pap smears regularly. You go get your pap smears regularly, it's estimated that 90 to 100% of sexually active people, not just women, 90 to 100% of sexually active people will have been exposed to some strain of HPV or in some form in their sexual activity lifetime. That means you probably had it. I'm not going to make you guys raise your hand and say who's sexually active, right? So I'm guessing most of us here have been sexually active at some point. You've probably had HPV. If you haven't yet, you will. Statistics say. Most of the time, HPV is inactive and your body will clear it just fine. But sometimes it leads to the development of cancer. Certain strains will lead to genital warts, and then certain strains will lead to cancer. And the ones that do not have symptoms, like the ones that cause cancer, are the ones we need to be worried about, okay? The ones that lead to cancer are not the ones that cause genital warts. So you can have HPV and have absolutely zero symptoms whatsoever. In fact, almost all men that have HPV will not have symptoms. So they're just spreading it around. Okay. You could be part of that. That's why protection, barrier, and things like that is so important. But that's also why pap smears are important, because you might get it and not know that you had it and clear it out and you'd be fine. But you might get it and not know that you have it and not be going to your appointments and not understand why you're not feeling well. Go to the doctor, find out you have stage 4 cervical cancer. And there's nothing we can do for you at that point. It's very actually pretty serious cancer once it gets to that stage. We still have people that die in the United States from cervical cancer. It's been drastically diminished by pap smears. So go to the pap smears, encourage your patients to go to pap smears. Just to be clear, if you've ever gone to a doctor and they're like, oh, we have some abnormal cells, we might want to keep an eye on these. Congratulations, you had HPV. That's what that means. It's just your doctor doesn't always tell you that that's what it is, okay? But it's so common that, I mean, it's common. It's not even reportable. It's not reportable. They don't have to tell the CDC that they've seen it. You know, it's that common. It's just as common as having like just regular warts or something like that, or a cold or something. It's super common. So just be aware of that. I'll be completely candid here. I've had HPV, just like you probably have. I'm even completely honest here. But I had abnormal cells as a result of that. And I had to go to get more frequent pap smears every 6 months. I went to go get pap smears and they would check on the abnormal cells. And when one time they got, I don't want to say out of control, but they did not get better, right? So we did colposcopy where they pinch off areas and they try to remove any areas that indicate there could be or any abnormal cells. And if it gets worse than that, they'll do a full biopsy where they'll go deeper into the tissue and remove those. But you want to do that before you get the cancer. And they can remove all those abnormal cells. And ever since then, that was like over a decade ago. Like we're talking like 20 years ago, I had this issue. I'm like 41 now, so I have had time, right? But every single pap smear I've had since those treatments have been fine, normal, no signs of HPV, anything. So your body can clear it out, but you don't want to let it get out of control if there is a chance for that. So keep an eye on it. Bacteriophages, again, they're going to inject their DNA into the bacteria and grow more. Here's an example of a bacteria that's getting attacked by just a ton of these guys. And once they make new, they burst out. There's 2 different cycles. We're almost done. There's 2 different cycles of these guys. They can be lytic or lysogenic. Okay, so I remember the difference like this. Lysogenic is whenever the viral genes become part of the bacteria genes. Okay, so when the phage genes become part of the bacteria genes, this is just bacteria, okay? When phage genes become part of the bacteria genes, that's a lysogenic cycle, okay? Whenever the phages are multiplying new phages and making the new phages, not hiding out, that's called the lytic cycle. And that's what leads to lysis or busting open of the bacteria. Okay? When we're making the new viruses, that's the lytic cycle. These phages that can switch between the lytic and the lysogenic, hiding out in the DNA or making new, these are called temperate phages. And just like we have the proviruses that hide out, like our HIV does, where that genome hides out in our genes, that's the provirus, they have prophages. It's the same thing. So when you have a prophage incorporated in your DNA as a bacteria, then you are you're going through that lysogenic phase of that, right? All right, so temperate phages do that. And if you want to start making a virus, you have to go through induction. You have to be induced to do it. All right, sometimes whenever you have a bacteria that's infected with a virus prophage, that prophage becomes part of their DNA. Sometimes those prophages come with extra traits like making toxins or being antibiotic resistant and other things like that that can help with survivability. Anytime that happens, that's called lysogenic conversion. We have converted that bacterial cell into being more pathogenic. Lysogenic conversion. So we're hiding a prophage DNA, carrying extra traits, and making our bacteria more infectious. This is not rare. Most of the pathogenic bacteria that cause diseases in our bodies actually is because of this. So this is not uncommon. It's actually a big deal, and it is one of the most common things that we're going to be talking about when we talk about viral infection of bacteria throughout the chapter. I'll talk a lot about lysogenic conversion, okay? Diphtheria, which we don't have a lot of anymore in the United States, but it still exists in the world. Diphtheria, cholera, don't underestimate some cholera, man, okay? Cholera, which is watery, rice-like diarrhea water. I don't know how to call it. Like, you will get so much bad diarrhea and release so many electrolytes in water that you'll be pooping what looks like rice water. Not brown poopy. I'm talking like rice water, like white, clear water. just continuously, and you will die from dehydration from this. We're talking about high mortality rates here. Okay? So that's cholera. And here's the treatment for it. Make sure that you drink fluids and electrolytes, and we'll pop you on a bed with a hole in it with a bucket under it. That's the treatment for cholera. Okay. So cholera toxin that leads to that, and botulism. So actual botulism, when you guys go get Botox injections, the toxin, the botulinum toxin, that toxin is actually coated by a phage that infected the Clostridium bacteria and gave them that toxin. Isn't that crazy? So it's become part of their actual species, but it's because of a virus that they have that. Viruses, obviously, they have to be in cells and stealing the equipment. So if you want to be a virus that's replicating and you want to replicate that virus to study it, you need to be using cells to grow them. You can't just grow them on agar. You have to give them cells to grow in, right? So that's tissue culture. We've already talked about a lot of these viruses and mortality rates, but your cells can actually make something called interferon to signal the cells nearby that there isn't a viral infection occurring. It's sort of like if you had somebody breaking into your house, you might not be able to help yourself from getting broken into and shot by the guy, but you can call your buddy next door and tell him to get his shotgun out. And he can have a shotgun ready at the door when the guy comes over, and he can save himself. That's sort of what interferon does for your cells. Causes them to start prepping. So that's it for the viruses. And just briefly, our non-cellular prions. Prions are literally just misfolded proteins. So if your protein is normally shaped like this, give me just a second. I promise we're just gonna go up to lab and get everything done. If your protein is normally shaped like this, and the blue part is hydrophobic, and we're hiding the blue part on the inside because it's hydrophobic, right? It doesn't interact well with the wateriness of your whole body. And this protein has a normal function in the body. Well, if it misfolds and now it's shaped like this, this isn't going to interact well with your body, right? If our hydrophobic, our water fearing is on the outside. So it won't interact well with your body. So those guys will actually, if they misfold like that, go near the healthy working protein, cause it to misfold, and then they'll just stick together because they can't hang out in the water, right? So this leads to a spongiform encephalopathy. Don't be afraid of the word. Spongiform literally just means it's going to make sponge-like holes in the brain. It's the brain because that's what encephalo means. Encephalo just means brain. Pathy means disease. So we're talking about sponge-like hole disease in your brain. That's all those words mean, okay? Because those proteins will aggregate like this. So we see a similar thing caused by aging whenever we talk about Alzheimer's and things like that. It's very similar to what we see with Alzheimer's, but it's a little bit different because that's just triggered by a natural process in the body. This is triggered by introducing a misfolded protein into your body. Okay, so what I mean is you go in the 90s and you ate a burger in England, right? Some beef that had misfolded protein in it. It gets into your body and contacts other normal proteins that are the same function in the human body, causes them to misfold like this one over here, and now causes a chain reaction in the body. We're talking eating a burger in like 1995, and 30 years later, this has happened to such a degree that now you have holes in your brain that 30 years later, you'll start presenting with some symptoms that look like dementia first. They look like dementia. There's nothing we can do for you at that point. There's nothing we can do for you once it starts happening. There's no treatment for this. It's just a misfolded protein. What are you going to give that person? There's no antivirals. There's no antibiotics. You just die. And we don't know it typically most of the time. We just think it's something else. And then we remove that person's brain and it's like Swiss cheese. And that's when you find out that this person probably had a spongiform encephalopathy. That doesn't sound familiar. What I'm talking about in that particular case would be mad cow disease. Mad cow disease is a prion disease. It's just a misfolded protein causing your other normal proteins to misfold. You can have genetic dysregularities that can predispose you to this, but there's nothing that like says you will get it right now. We actually don't know for sure what makes more people, some people are more susceptible than others. It just seems like it might be genetically related. Anyways, it takes a long time. And this is another 100% fatal one, but it's not a virus. It's just a misfolded protein. And it's actually really hard to control these guys. This led to the culling or killing of like thousands and thousands of cattle at the time. And if you feed cows, that's how they were getting it. They were just grinding up dead cows and feeding it to the cows because they don't have enough land. Anyway, so that's a misfolded protein in humans. We call this Creutzfeldt-Jakob disease, CJD. This can happen naturally in humans. That's just normal Creutzfeldt-Jakob disease. If you get it from mad cow disease, they call it variant or VCJD. So do be aware of what CJD is, because that will be on the test, okay? I won't make you spell it, but be able to recognize it, okay? Creutzfeldt Jakob disease. There's other ones as well. If you guys have ever heard of chronic wasting disease in deer, and now it's spreading to elk. So that used to only be for deer, and now it's spreading to other species, which we didn't think was possible. It hasn't spread yet to people, but that doesn't mean that it can't. And so they track that. If you're a hunter or you know somebody that hunts, they're supposed to bring samples to get tested for chronic wasting disease. We haven't typically had it in Oklahoma very much. We've had a couple of cases, but it doesn't seem to be endemic here yet. But it is something that is rampant in the United States as a whole. So that's a prion disease. There's also ones like Kuru, which is something that exists in human cannibals. Which, yeah, are you going to see that? Probably not. But they venerate their dead by eating them. So they believe that having the person in your tribe that dies, making that loved one become one with the whole tribe as a family. But then if they died from prion disease, they're just spreading it on throughout the whole tribe. That's called Kuru. There's also fatal familial insomnia, which is a misfolded protein. It becomes misfolded due to genetic relation, like something to do with your genetics. But it becomes misfolded and you can never sleep again. And you die from not sleeping. Yay! It's cheerful, isn't it? I love it. I think it's so exciting. But that's it. So we're done. Our questions just really quickly, and we'll go right up to lab. Essential components of a functional virion, the two things you absolutely need, the capsid and the nucleic acid genome. What is the term for viruses that lack an envelope? Naked. Viral infection, those 6 steps were adsorption, penetration, uncoding, synthesis, assembly, and release. Makes sense? And of course, I gave you the 6 steps. It should be pretty easy to put them in order, right? Something like that. Where do viruses with the DNA genome replicate? In the nucleus, and it's DNA nucleus. And what is lysogenic conversion? When a prophage, which is a phage DNA in the bacterial DNA, confers new genes to the bacteria that help it survive better. Okay, that's it. Let's go up to the lab. We got carried away in discussion. That's my fault. But everybody gets excited about a virus.
It's not like you're not putting in your own virus.
I know.